Good evening, everyone. It gives me immense pleasure to welcome you all to today's webinar on behalf of the Center for Scientific Research and Development, People's University, Bhopal. Welcome to yet another exciting edition of the BioConnect webinar series, a platform led by students, for students, designed to ignite curiosity, foster meaningful dialogue, and connect academic knowledge with real-world challenges. My name is Uttam, and I am proud to tell you that I am a MSc Biotechnology student. I am honored to serve as your MC for today's session. Today's webinar focuses on the important theme, from containment to cure, the critical role of BSL-3 labs and organized infectious disease research. In an era marked by the rapid emergence and re-emergence of infectious diseases, ensuring safe and effective research environments has become critically important. As at level 3, IMNPSL-3 laboratories play a vital role in handling highly infectious pathogens under controlled conditions, protecting both the researchers and the environment. At the same time, the use of organized, miniaturized, lab-grown models of human organs has revolutionized disease research by enabling more accurate study of pathogen interactions and drug responses. Together, BSL-3 facilities and organized technologies are accelerating the development of targeted therapies and improving our ability to respond to global health challenges. Before we proceed further, I would like to extend my heartfelt appreciation to the leadership of People's University, our respected faculty members, and the dedicated student team at CSRD, whose efforts have made this event possible. We are deeply honored to have several distinguished personalities joining us today. I warmly welcome Honorable Vice Professor Dr. Harish Rao, Vice Chancellor of People's University, our guest speaker, Dr. Jayashree Garyan, who is Director at Stanford University School of Medicine, Stanford, California, USA. Professor Dr. Subhash J. Bhore, Head of the Center for Scientific Research and Development and mentor for this initiative, and Ms. Akanksha Lodhi, Chairperson of the Organizing Committee. We sincerely thank all guests and participants for joining us today for this enriching webinar session. To formally set the stage for today's webinar, it is my honor to invite Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, to deliver opening remarks. Sir, over to you. Please proceed.
Thank you, Mr. Uttam Singh. Good evening to everyone present here and to everyone who have connected online for this webinar. Distinguished speakers, respected faculty members, dear students and participants joining us from India and around the world, my warm greetings to all of you from People's University. It gives me immense pleasure to welcome you to BioConnect webinar number 16, a wonderful initiative under the banner of the Center for Scientific Research and Development, CSRD for short, under People's University, Bhopal. I am particularly delighted to note that this is a student-led program reflecting the spirit of innovation, ownership, and academic curiosity that we strive to nurture at our university. Today's topic, from containment to cure, the critical role of BSL-3 labs and organoids in infectious disease control, is both timely and significant. In an era where infectious diseases continue to challenge global health systems, the role of advanced containment facilities such as ESL-3 laboratories, along with cutting-edge models like organoids, has become indispensable. These platforms not only enhance our understanding of pathogens, but also accelerate the development of diagnostics, therapeutics, and even vaccines. I am confident that today's session will provide valuable insight into how science is evolving to address some of the most pressing health concerns of our time. It will also inspire our students to think beyond textbooks and engage in translational and impactful research. It is my great honor to welcome our esteemed speaker, Dr. Jayashree Garyan, Director at Stanford University School of Medicine, USA. We are privileged to have a distinguished expert of her caliber share her knowledge and experience with us. I am certain her talk will be enlightening and thought-provoking for all participants. I would also like to appreciate the efforts of the organizing team from PSRD for planning and executing this session under For the Students, by the Students initiative. My sincere congratulations to all student organizers for their dedication and enthusiasm. I also want to thank Professor Subhash Bore, Head of the Center for Scientific Research and Development, for his constant guidance, mentorship in shaping this academic platform so smoothly over the past 16 episodes. To all participants, I encourage you to actively engage in today's session, ask questions, and make the most of this learning opportunity. Such platforms are invaluable for expanding our academic horizons and building meaningful scientific perspectives. With these words, I once again welcome you all and wish the webinar great series, and I'm sure you'll have an immersive learning experience. Now I will hand it over to our MC, Mr. Something. Over to you. Thank you.
Thank you very much, sir, for your inspiring words. Now, I invite the chairperson of the organizing committee, Ms. Akanksha Lodhi, to deliver the welcoming remarks. Ms. Akanksha, over to you.
Thank you, Mr. Uttam. Hello, everyone. Our esteemed guest speaker, Dr. Jayashree Garyan, our Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, respected dignitaries, our esteemed faculty members, our mentor, Professor Dr. Subhash Bore, and my dear fellow students and participants. My name is Akanksha Lodhi, and I am proud to share that I am a student of MSc Microbiology at the Department of Biotechnology and Microbiology, Center for Scientific Research and Development, People's University, Bhopal, India. On behalf of the organizing committee, it is my great privilege to welcome you all to the BioConnect webinar session number 16, a special student-led event organized by a dedicated team of biotechnology and microbiology students at the Center for Scientific Research and Development, People's University, Bhopal. Today's webinar, titled From Containment to Cure, the Critical Role of BSL-3 Labs and Organoids in Infectious Disease Research, addresses a highly relevant and impactful topic that lies at the intersection of innovation, biosafety, and infectious disease research advancements. We are deeply honored to have with us our distinguished speaker, Dr. Jayashree Garyan, Director at the Stanford University School of Medicine, California, USA. I would also like to express our sincere gratitude to our Honorable Vice Chancellor, Professor Dr. Harish Rao, for his constant encouragement and for graciously joining us today to share his opening remarks. I also want to thank to our mentor, Professor Dr. Subhash Bore, for his invaluable guidance, encouragement, and continuous support in making this event possible. I would also like to acknowledge and congratulate to my fellow student organizers. Their teamwork, creativity, and dedication truly respect the vibrant spirit of People's University. With great pleasure, I once again extend a warm welcome to all participants. I encourage everyone to actively engage throughout the session as we look forward to an inspiring and insightful discussion. Now, I would like to hand over the program to our MC, Mr. Uttam. Over to you. Thank you.
Thank you very much, Ms. Akanksha, for your insightful welcoming remarks. Dear participants and fellow students, we are now ready to begin the core session of today's webinar. At this point, I am pleased to hand over the session to our moderator, Ms. Aisha, who will introduce our guest speaker and moderate the session. Ms. Aisha, over to you.
Thank you, Mr. Uttam. Good evening, everyone. Welcome to the 16th session of the BioConnect webinar series. I'm Aisha Zamir, and I'm proud to tell you that I'm a first-year MSc Biotechnology student at the Center for Scientific Research and Development, People's University, Bhopal. It is truly an honor to serve as the moderator for today's enlightening session. For today's session, our distinguished speaker is Dr. Jayashree Garyan. It is my honor to introduce the speaker, Dr. Garyan. She is a director at Stanford University, School of Medicine, California, USA. She leads a BSL-3 multi-user facility at Stanford University, overseeing scientific direction, operations, biosafety, and business strategy. With a PhD in biotechnology and infectious diseases from Jawaharlal Nehru University, Delhi, and a master's degree from Devi Ahilya University, Indore, she brings a globally informed and deeply interdisciplinary perspective to high containment research environments. Her career spans advanced research at Harvard University, Forsyth Institute, and the University of Massachusetts Amherst. She has built and led biosafety and operational programs, supporting over $200 million in grant-funded research. Passionate about advancing the vision of biosafety, decoding for practical applications, she ensures that regulatory compliance and scientific innovation evolve hand in hand. Beyond her institutional leadership, Dr. Garyan is an active member of UPSA International, contributes to the Distance Learning Committee, and engages with broader scientific initiatives through the National Science Foundation. Her commitment to research, education, and biosafety policy makes her a true leader in the field. With such an impressive background, we are truly privileged to learn from her insights today. Her talk will shed light on the critical role of PSL-3 labs and organoids in infectious disease research and how these innovations are shaping the path from containment to cure. With this brief introduction, it is my great pleasure to invite Dr. Garyan to deliver her talk on From Containment to Cure, the critical role of BSL-3 labs and organoids in infectious disease research. Dr. Garyan, over to you.
Hello, everybody. I'm going to share my presentation. Please bear with me. All right. So can everybody hear me?
Yes, we can hear.
Okay, perfect. So I am truly thankful for everybody here, the organizing committee, the Vice Chancellor, and Vice Chancellor Professor Dr. Harish Rao, the head of the institution, Professor Dr. Subhash Bore, and the organizing committee and all the participants to be here at this time in the evening. I know it's evening time in India, and it's really early morning in California here. So, and I am excited because you are from my state. I basically grew up in Indore and doing schooling there, and then went on to do PhD, master's. I also did master's in North Carolina in physiology, and now I'm here. So it's really, I'm more passionate about giving back to my own country and my own state, particularly because this is in Bhopal. So as a child, I was in Bhopal multiple times. So without further ado, and I'm going to start my presentation. As you all have heard, that I'm going to present an overview of containment to cure and the critical role of BSL-3 labs in organoids and infectious disease. So this is going to be the agenda for today. I'm going to talk about pathogens and search overview. Many of you might have seen pictures of BSL-3, but I'm going to introduce you to the BSL-3 concepts and why and what of BSL-3, a little bit delving deep into the BSL-3 levels, risk assessment, hierarchy of controls, PPE, that is personal protective equipment, via risk assessment. And then finally, I'll touch base on the new development that we are doing, which is in organoid research. So next, I'll move on to introducing the pathogens which cause the diseases, right? So there are highly infectious diseases which can happen from BSL level 1 pathogen to 3. And the focus of today's talk is the pathogens which are operated inside the level BSL-3 laboratory. And we're going to talk more about what is BSL-3 so everybody is on the same page. So here I have highlighted few pathogens which are operated inside the BSL-3 laboratory. Bacterial pathogens, it can be viral pathogens and fungal agents as well. So among the bacterial pathogens, I know in India there are certain laboratories which are working with select agents called Bacillus anthracis and Brucella here. And these cause a disease called anthrax. Brucella causes brucellosis. Other bacteria like Yersinia pestis cause plague. Francisella tularensis cause tularemia. Burkholderia malayi cause glanders, and Coxiella burnetii cause Q fever. In the viral category of pathogens, there are multiple viral pathogens which keep haunting us. And the recent one that everybody knows about is SARS-CoV-2, which has caused the pandemic. The other one is SARS-CoV-1, which is a select agent. And another coronavirus that I work with is MERS-CoV-2. So these all cause infectious diseases of lungs. And also SARS-CoV is now known to be causing long COVID even after pandemic has gone. The next one is hantavirus, Nipah virus, yellow fever, West Nile viruses, and so on and so forth. We know that in India, chikungunya virus is quite prevalent. In US also, people have started working on chikungunya virus. Now, recently, we have known that H5N1 and monkeypox virus have caused outbreaks. And so they are recently main concerning pathogens. In fungal agents, also, we would include multiple fungal agents, including Coccidiosis imititis and Histoplasma capsulatum. So I would mainly focus on bacterial and viral pathogens because fungal pathogens are there, but there are not so many in the BSL-3 category. So moving on to the next slide. So I'm going to delve upon Mycobacterium tuberculosis, which is one of the bacterial pathogens that is from the very beginning always been a BSL-3 pathogen. And it is very prevalent in India and on those regions. It's basically endemic. And it has led to almost 1.23 million deaths worldwide. And in this right panel, you will see the electron microscopic image of the Mycobacterium tuberculosis. As you can see, it's a long anaerobic bacteria, and it is an acid-fast and non-motile bacteria. It causes latent tuberculosis, pulmonary tuberculosis, and extra pulmonary tuberculosis. And the reason it is worked inside the PSL3 lab is because the infectious dose for Mycobacterium tuberculosis is less than 10. That means even 10 Mycobacterium tuberculosis, which is very low, can cause, if you're exposed to that many number, it can cause diseases. And another reason is there's no effective vaccine for adults whatsoever for this pathogen. So these two combined makes a good case for high-risk pathogens. So they need to be only dealt inside the PSL3 laboratories. In some countries like India, BCG is provided to children for the effective protection, but as the children grow, they don't provide any protection. The BCG does not. However, in US, there is no BCG vaccine provided to children. So next slide. I will now switch to virus. There are two viruses that I'm going to talk about. One is the MERS virus, which is Middle Eastern Respiratory Syndrome. The name it is called Middle Eastern because the origin is in Middle East. How it spreads is the MERS-CoV-2 virus goes from, infects the bats, and it's a zoonotic disease. So it goes from bats to camels, and from camels, it gets zoonotically transmitted to humans. Anybody that is coming in contact with a sick camel who is infected with MERS, they can get infected with, those people can get infected with MERS. And as you can see in the left panel, the once you have the infection, it can spread to anyone and everyone you get in contact with through aerosols. So the human route of transmission is also aerosol, just like Mycobacterium tuberculosis. The infectious dose is again very low, and MERS is very scary compared to MTB because it has no vaccine at all, and the therapeutics are also not that developed. So moving on to SARS-CoV-2, which most of you and all of us know, it has become a household thing. In the left panel, you can see that there is a picture of SARS-CoV-2, the electron microscope picture, and alpha gamma variations are there. So it has caused over 103.4 million deaths, and cumulatively, it has caused, the cases is 103, and the deaths are 1.2 million. Again, the human route of transmission is droplet or aerosol. The incubation period takes 1 to 14 days for symptoms to appear. And there are various symptoms which are very similar to a flu. And there have been, because of the pandemic, there have been there's multiple vaccines available, and there are also a lot of drugs being developed. So my facility, we played an important role in developing some of the drugs which are right now under clinical trials. So the point of having all these three pathogens explaining to you in detail is that all these three pathogens, whether bacterial or viral, they're highly infectious. There are no vaccines for them except COVID, and the therapeutics and the drugs are very limited, right? So any kind of disease which is so infectious and they have no drugs or vaccines available or very limited available, what research can be done on those? So the research can be like for an unknown pathogen which appears, we can do immunological studies. So in BSL-3, you can work on these agents, these pathogens, and develop the strategy, because you can only know the strategy after you have known the immunological response. So in the right panel, in the beginning when this pandemic hit, we did not know how SARS-CoV-2 works, how does it bind to cells, how is it internalized, and what is the immune response. So we started studying the immunological response. The entire community of scientists started immunological response study. So found that there are two receptors, essentially, the ACE2 and TMPRSS2, that bind to the virus and internalizes the virus to the cell, and then so on and so forth. We get various immune reactions. First is the immune response, which is to the cells, which cause, if the person is not able to handle it, then it is a leaky response. And in the right panel of this image, you can see that if a person is able to have immunity because of the having the vaccine, they can react and produce more antibodies and provide protection through CD4 and CD8 cells. So this is one of the areas which can be researched on, and we are researching on this. The second is after understanding the immunological response, we can discover the new drug candidates, which can be more effective and more efficient. So, because without drugs, we cannot even start to handle the infectious diseases. So here, for example, on the right panel, you can see that in the dish, we use the in vitro technology where the cells are infected by SARS-CoV-2 or any pathogen for that matter. And then we can see that how the infection is growing through immunoluminescence assay. And after adding antibody or drug, here I'm representing drug as an antibody, and then they can bind to the cells and provide the infection can be low. So these are the kind of immune drug assay we developed in the BSL-3 labs. So the next research that is being constantly done in the labs for tackling the infectious diseases, developing the new diagnostic assays. So these assays require patient samples and tissues and manipulations of those samples and tissues. And so this can be infectious and dangerous to be held in the labs which are below BSL-3. So these patient samples and tissue samples are handled inside the BSL-3 many times, depending on the procedures, and then use proteomics, genomics, or regular binding to develop assays which can help in diagnostics of these diseases. So recently, like, we are developing a Mycobacterium tuberculosis diagnostic assay, because in the labs, it takes a long time to grow Mycobacterium tuberculosis. So if you take a sputum sample from patient and grow it in a lab, then it takes almost 1 month to grow. So it takes a long time to diagnose. So to shorten the time, there is a new diagnostic assay that is being developed currently in the lab, where it can be linked to the luminometer, where instantly you can test the sputum in a luminometer after various steps to see whether the patient is positive or negative. So these kind of development assays has to be first done in BSL-3 and then optimized and then taken out in the bench. So these are the few publications that we were able to do. The first one is pertaining to, like I said, the immunological assays. The second one is for drug development, like I said, and then the last one is for organoid one. So we're going to talk about more organoids in the later section of the presentation. So let's now dive deep into the BSL-3 levels. So what are BSL-3 levels and how do we define that which level is what? So biosafety level 1, most of the labs, they're working in biosafety level 1, where the disease, the bacteria or the pathogen that we're talking about is not known to consistently cause the disease in healthy human adults. And there is a minimum potential hazard. Examples of BSL-1 category pathogens are Saccharomyces cerevisiae, E. coli K12, and non-infectious bacteria. The BSL-2 level labs, however, have moderate potential hazard to personal and environment. So the bacteria example for this is hepatitis A virus, Streptococcus pyogenes, Borrelia, and which causes Lyme disease, and Salmonella species. So the next one is BSL-3, which we are talking about in this presentation. The microbes that are handled inside BSL-3 can be either indigenous or exotic. They can cause serious or potential lethal disease through respiratory transmission. So the examples we have cited a lot in the beginning of this presentation are cited here again. The Eiserinia pestis, Mycobacterium tuberculosis, SARS, rabies virus, West Nile virus, and hantavirus. However, I would like to note here that NIH has downgraded SARS-CoV-2 from BSL-3 to BSL-2 because we know now that there are vaccines which works for SARS-CoV-2 and also there are drugs that are available. So many universities have chosen to downgrade the SARS-CoV-2 work from BSL-3 to BSL-2. Now there is another level which is called BSL-4. Now, what are these? I have been to several BSL-4s and maybe like 10 BSL-3s, and I've worked in 10 different BSL-3s. For BSL-4s, it is related to dangerous and exotic, posing a high risk of aerosol transmitted infections. And infections caused by these microbes are frequently fatal. That means they immediately cause death and they have no treatment or vaccines. So these kind of pathogens are only handled in BSL-4. Examples are Ebola virus and smallpox viruses. All right, so now we go in detail on BSL-3. What is BSL-3? Does it require special facility design? Yes, it does. It requires a special facility design where there is a negative pressure maintained inside the lab. That means whenever you open the door of a BSL-3, all the air from outside goes in. That means there is no possibility of escaping of these viruses or pathogens outside the lab. As you can see in the left panel, there are arrows for the air to go in, and then the air is circulated here, and then it is exhausted out of the building or the room through HEPA filter. So there is no danger to the environment as well because air has to come in and get out, right? There has to be continuous flow of air, otherwise people won't be able to work there. And so the air is filtered out and then it is released in the outside environment. Other features of the facility are the windows are sealed, there is a hand washing sink which is required, and so there are multiple facility design which are involved in BSL-3 facility. In the right panel, I'm showing you my lab, which is a part of my lab where it is showing the directional flow. So this is the lower panel in the blue is outside of the BSL-3. It's a corridor, and the red arrows are showing the direction of the airflow. So the air is going from outside to inside. And so there is no chance for the escape of the pathogens to go out whenever the door is open for entry and exit. And there is also negative pressure. So -1 is the pressure in this room, and as you go and enter the second room, it's -2. So this is just a number here, but actual numbers are different. It is based on the water pressure. So this is explaining how the facility design is different from the BSL-2. In a BSL-2 level labs, you don't need negative airflow, you don't need a HEPA filter exhaust, you don't need a directional flow, you don't need sealed windows, and hand washing sink is required, but rest of the things for the BSL-3 facility is not required in BSL-2. So moving on, what are the next level that are required in BSL-3? The next requirement is physical security and controlled access and a visitor policy. Not everybody can just enter the BSL-3 labs. Even if you are part of the university and you have card access to all the labs, BSL-3 needs to be having a physical security with keys and controlled air access. Only people who are trained and only people who are authorized to go inside can enter the secured building or the secured lab. So now with this little bit of overview on what are the BSL levels and what are the BSL-3 labs actually required. After this, what is the risk assessment and management to work with these pathogens? We're going to talk a little bit about that. So it depends what kind of pathogen you are working on, right? So it can be bacteria, virus, or fungus. And then what procedures you are actually doing. So are you pipetting, which of course many people are pipetting whenever you're dealing with research in the pathogens. And are you doing centrifugation? Are you using syringe and needles? Are you doing sonication? All those procedures are very important to do the risk assessment. So first of all, identify the hazard, right? Once you know the hazard, and then you know the hazard, how do you identify the hazard is what, where, and how the work is done, who is involved in working, and what can go wrong. So this risk assessment is a very involved process, and it requires scientists and all the stakeholders from the university to think about the risk assessment. So it is basically conducted by identifying the hazards and what are the risk occurrences and consequences severity. There is a 5x5 matrix that is developed by the scientist or the occupational health or EHS, and they figure out the risk of that pathogen and the risk of that procedure involved with the pathogen. Because the pathogen might be very highly infectious, but the procedure would be very low, you're just pipetting and not doing anything else, then the risk assessment would be different. Versus if the pathogen is infectious, is not that infectious, but you're doing a very highly risky procedure, like you're using syringe, using needles, you're using sonication, then the risk assessment is different. Okay, and then also how you manage the risk. First, you find what's the risk level. Is it low, moderate, or high? After doing risk assessment, what do we get? We get whether the whole process is low risk, moderate risk, or high risk. Once you identify the risk, then we move on to doing the risk management, right? So how do you manage the risk? So for this, you already have identified the hazard and risk. The next is to evaluate how severe can it, if there is an exposure, how serious is the exposure? And if there is an exposure, what is the risk of that exposure? Is it very minor? Is it acceptable or unacceptable? The next one is we determine the controls, how we reduce the risk. We say that, okay, this is not acceptable that anybody gets infected with my Mycobacterium tuberculosis in the lab. So how do we determine that we can reduce the risk for this kind of exposure? The next is implementing controls. So what can we do? Can we do a medical surveillance program? Can we vaccinate everybody if the vaccine is available? And stuff like that. The next one is review the effectiveness. Once we have risk assessment and a risk management process in place, then the next step is annually review it, because everything changes every time, yearly basis or monthly basis. So we have to always be updated and review the effectiveness of controls. So no matter what we do, if we are working with a very highly infectious disease pathogen, then we cannot zero the risk. Anything we are working in the research, we cannot zero the risk. We can minimize the risk, right? So how do we minimize the risk? So this is the hierarchy of control, which helps us decide how to minimize the risks. And this is used in all the universities and all the institutions who are working with pathogens or chemicals, or for that matter, any recombinant DNA technology related single-stranded or double-stranded RNA. So for here we can see in the left panel, there is an inverted triangle. And on the top of that triangle is elimination. That means you can actually zero the risk if you eliminate the pathogen. You say that we are not going to work with this pathogen because it's too risky. So we zero the risk. But researchers want to do the research, so they want to use it. So what would be the next if they say we cannot eliminate the research? So next, what we'll do, we will substitute. So in green, you see substitution. That can we use attenuated strain, which is less risky, which is less infectious, and which does not cause the disease. So after elimination, if that doesn't work, then the next step is we substitute with a low infectious pathogen. Then the next step is after substitution. If that happens, it's okay. If it doesn't, then we move on to engineering controls. So what are the engineering controls? Engineering controls are something that isolate people from the hazard. So what are the hazards here? Hazards is the pathogen, right? So how do we separate the people, that means the researcher, from the hazard? So on the right side, you can see the very right side, there is a biosafety cabin. That is the best way to isolate people from the hazard. You work everything that you're manipulating with pathogens, any research that you're doing, any experiments you're doing, do it inside the biosafety cabinet. And that there is a sash between you and the biosafety cabinet. There's air circulation which goes inside and it gets filtered. So there is an isolation of people from the hazard using engineering controls like biosafety cabinets. Next is administrative controls. So once we are done with our engineering controls, we can do apply administrative controls. And what are administrative controls? Administrative controls means developing the standard operating procedures. Like you are pipetting, how should you pipette? Should you pipette slowly or very vigorously? If you pipette vigorously, then the chances of aerosol spreading can be more. Do you work slowly or very fast? Then everybody, the SOP is developed that everybody working with BSL-3 pathogens should work very slowly inside the biosafety cabinet, because if you do rush, then there can be chances of spill and the pathogens and your tubes can spill out of the biosafety cabinet. These are the very basic administrative control. There are more administrative controls, like you can only operate centrifuge in certain way, you can only operate the sink in certain way, you can only do the shaking in certain speed because higher speed can cause spill and all those kinds of things. The next and the most popular hierarchy of control is PPE, that is personal protective equipment. So you protect the person or the researcher from the pathogen by providing them personal protective equipment. What are personal protective equipments? We're going to talk in detail about PPE because these are the most popular and the most safest way other than the administrative controls and very important. So on the right panel, you can see that double gloves that the researcher is wearing, face shield, goggles, solid front gown or suit. Those are the PPEs, and also eye shield and boots. So those are the PPE along with gloves that are required for the personal protective equipment. So on the right side, on the very right side here, you will see two long columns which are showing that most effective to least effective. What is the most effective way to reduce risk? The most effective is to eliminate, right? And what is the least is PPE. And then the second one is least supervision required. For elimination, you don't need any supervision, just eliminate the pathogen from your research work. And the most supervision is required as we go down. We need to see if everybody is trained properly for PPE, everybody is working properly, and engineering controls are in place. So this is the hierarchy of control. We're gonna delve deeper inside in these topics of PPE. So your team and your groups of students are mostly master students, I realize. So you deal with doing research in the lab, maybe lab BSL 1 lab or 2 lab, and you have seen all these PPE, at least you have used one or two of them in your lab experience. So you can see I'll start from the very basic, from the right, gloves, right? All of you have used gloves. So as we go in the higher risk category, we use double gloves, the inner gloves and the outer gloves. But mostly in the BSL 1 and 2, depending on the risk, you only wear single layer of gloves, right? The next is eye protection. So eye protection, what it does is, if you know that it helps you from splashing, and it protects you from splashing anything in your eye when you're working with a research pathogen. So if the risk is very high, then you wear face shield. It protects your entire face. If risk is less, then you can do goggles, just simple goggles. Then I will move on to this one here, which is protective clothing. You all are aware of the gowns, like lab gowns. Many of the lab gowns, depending on, are disposable or cloth, and they can be of various kinds. They can be solid front, they can be scrubs, which are worn in the hospitals mostly, but they can also be worn in the lab, and then they are left in the lab. The next one is full body suit or coveralls. So depending on the risk assessment, the PPE can change in the lab of BSL-3. The next is shoe covers. You mostly want to wear shoe covers in the lab, so you can dispose your shoe covers and not the shoe. And then respirators finally. So it's important because all these pathogens are aerosol transmitted. It's important to have a respirator in the BSL-3 in spite of it having a negative pressure. You need to protect yourself from respiring any pathogens that might come in contact because of the manipulations. So the most basic is N95. Most of the BSL-3s who are dealing with mycobacteria or any pathogen, they can use N95, but if the risk by the institution is considered high after risk assessment, then they use something like this. this equipment, which is called PAPR or CAPR. So very high aerosol generating procedures are risky, and so these are used, and they are powered by batteries, which is shown here. However, the N95 is just a simple N95, and then it can be used in the laboratory for many purposes. For simple microbiological practices, N95 is used. How also we determine which one to use is basically based on the risk assessment, whether it's low, medium, high. Secondly, it also depends on the resources that you have. Some people might have financial resource constraints, so then you use things like N95 and also solid front count and the coveralls, right? So moving on. What is also required along with PPE and along with all the administrative control and engineering controls, which is very important to protect the researchers. It is a medical surveillance program. So medical surveillance program is offered by the institution if you're working with the BSL-2 or BSL-3 level pathogens. First of all, it includes fitness for duty. So the doctors and the medical surveillance program will check the researchers for the fitness of duty, whether they're fit for working in the lab and when they're exposed to all these pathogens, because there is a risk involved. For example, if the vaccine is available, then they're vaccinated. For TB, since the vaccine for adults are not available, there's a regular periodic TB screening. So it can be annual TB screening. So whoever is working in the TB program for TB research are screened on an annual basis for TB or on 6-monthly basis, depending on what your institute decides. And the next is respiratory protection program. So as we saw in the previous slide, that there is a need of a respirator. That is either it can be N95 or it can be a PAPR or a CAPR. So enrolling in that program is important in United States over here, because they want to make sure that you are fitted with the right kind of respirator. All right. So now, with all this in mind, anything bad can happen, right? Any emergency can happen while you're working in the lab. So how to protect that? Protect you from that, protect the researchers and the lab from that. So that requires an emergency management program. And what all can happen? There can be a natural disaster. There can be flooding, which can flood the lab. There can be an earthquake. There can be a building or facility emergency, which is like fire or something. There can be a medical emergency that the researcher is nauseous and they fall down. There can be a spill meaning anything that is you're handling, the liquid culture can spill in the floor and it can cause an emergency situation. And that can also cause potential exposure. So, like I mentioned, a natural disaster, earthquake, and flooding. For building emergency, it can be plumbing malfunction, it causes flooding, it can be fire, it can be pressure alarm. We already discussed that in the biosafety level 3, there has to be a negative pressure in the lab. But because the building malfunctioning, the pressure can be lost, and that can cause exposure. There can be a power loss or electric loss, right? So if there's a power loss, none of the biosafety cabinets will work, and the pressure alarm will also not work. And one of the other malfunctions can be your biosafety cabinet, which is providing you a barrier from the hazard, can start malfunctioning. So all these needs to be taken care of, and an emergency management program needs to be developed. That what if this happens? Do we have a fallback plan? How do we exit the lab? Those kind of things. Your autoclave, which is very important to dispose of, the materials that you're working with, and also to sterilize the material can malfunction. The other unknowns are there can be a theft. A person can come and try to steal or steal the biosafety pathogens and cause a biosecurity issue. An intruder can come. So the next on the right panels are medical emergency, which is a person who's working with the research in the research environment can be conscious, but they fall down. And the second can be they can unconscious. So in both these scenarios, how do we handle that emergency? And specifically, these are very challenging when this happens inside the BSL-3, where the 911 or the emergency first responders cannot go inside because there is a threat to their lives as well. So for the spill, it can be either inside the biosafety or outside the biosafety cabinet. Or it can be inside any equipment, say incubator or centrifuge. So how do we handle that spill? Now exposure can be biological exposure. We've been talking about biologicals a lot here in this talk, but there can be a chemical exposure as well. So all these points for emergency management needs to be taken care when you're working in the BSL-3, for that matter, even BSL-2 and 1, right? But why do we discuss it here? Because it becomes more important. Because any of this happens and the pathogen escapes, it's a biosecurity issue. And also it's a big bioexposure issue. So all these have to be listed in your biosafety manual. There has to be a biosafety manual which lists all these procedures, that what we should do. This is another talk from another time to go in detail about the emergency management and what should be done. If there is a spill, here's a picture in the middle to show if there's a spill, how you clean up the spill in the BSL-3. And so this is the section for emergency. Now I will switch gears and I will talk about organoids. There are only few slides for organoids. And let me know if I'm going over time, please, Dr. Subhasbor. And so for organoids, what are organoids? Organoids are 3D cell culture systems, right? They're actually cell culture systems which are growing in 3 dimensions. They can be called mini organs, basically, and they mimic some of the key multicellular, anatomical, and even functional characteristic of real organs. So in the image, you can see that there can be various organoids. It can be cerebral, like brain organoid, lung organoid, mammary gland organoid, liver, stomach, intestinal. So basically, any organ can be developed as a mini organ in the dish. Especially in this case, we have worked with cardiac organoid, that is heart organoid, also lung organoid and brain organoid. So what are the applications? So organoids can be developed by either cells or tissues. We mostly focus on tissue organoids. So what are the aspects of organoids? We can develop cancer therapies with organoids, using for solid and liquid tumors. We can deal with autoimmune diseases like celiac disease using the organoids. And we can also develop drugs and models for infectious organisms like virus and bacteria, here SARS-CoV-2. We can work with degenerative diseases like Alzheimer's. As you can see, there is a very big difference between healthy brain and Alzheimer's brain. So all these can be modeled in the dish. And the next one is allergy. If you have food allergy or skin allergy, those models of skin can be developed in the dish. So Organoids have various applications. We're going to focus more on the infectious disease application because that's the topic of today. We can have a normal or a diseased human tissue in the dish. So it depends on the source of the tissue. If you are getting uh, the brain or lungs from a diseased human, then you can develop a diseased tumor mini organ in the dish. Otherwise, you can develop a normal one. So last few slides. So when SARS-CoV-2 was very new, we did not even know that how the lungs are getting infected, whether, and so instead of going to the animal model, people developed a lung model. And they found out that lungs can be infected in the dish by SARS-CoV-2, and the models can be developed. Once you develop a model in the dish, then various drugs can be used to treat. But it was a big finding that we can develop an organoid model in the dish for SARS-CoV-2 infection. And here the pictures are showing that in the red, if you have the red, that means that organoid is infected with SARS-CoV-2. And SFT, PC, and DAPI. DAPI is for nuclei staining, and SFT is for the cells of the lungs. So these images are showing that successful organoid model for disease in the dish kind of model. Like, can you study a disease in a dish using the organoid model with SARS-CoV-2? Okay, next is we also developed a human lung organoid model, which is called ALOS, and which is an interface between air and liquid interface. The other one is also ALI, which is air-liquid interface. So there are two models we developed, ALOS, which is adult lung organoid, another is ALI. So these are two different models. We found that in the no drug condition, the infection of SARS-CoV-2, of course, is showing very high, versus we used a control drug, which is known, which is DS41524. The infection was very minimal. And for a new drug that we were developing, SFT3557, also it worked very beautifully and it decreased the infection. However, the infection was not low as control one, but it was still very significant. So these organoid models can successfully be used for drug treatment and instead of using the animals. The reason and the significance of organoid model is recently regulatory agencies and funding agencies have declared that they want to minimize animal work. So they want to see organoid work and as a substitute for animal work. So this is very helpful. And the high throughput is more with this. So with this, I would like to thank you all for inviting me for your presentation of BioConnect, and I'm happy to take any questions. Thank you.
Thank you, Dr. Garyan, for that highly insightful and thought-provoking talk on From Containment to Cure, the Critical Role of BSL-3 Labs and Organoids in Infectious Disease Research. Your presentation has beautifully highlighted the intersection of advanced biosafety infrastructure and cutting-edge biological models in addressing infectious diseases. I'm sure our audience has gained valuable perspective on both the challenges and opportunities in this field. Now we will move into the question and answer session. I encourage all participants to type their questions into the chat box. We will do our best to address as many as possible within the time we have. To begin, let me ask you a few questions on behalf of our participants. The question is, considering the high cost and complexity of BSL-3 laboratories, how can developing countries like India strategically expand such facilities to strengthen infectious disease research capacity?
Yeah, thank you for that question because I worked in BSL-3 in JNU in India. I faced the low resource problem and versus in US, there is always a funding issue, whether you are in a low resource country or in a high resource country. But BSL-3 is another demon and another elephant. It requires constant input of finances. And so there has to be a mindfulness about how the program of BSL-3 is in the low resource country, especially with, the PPE, what kind of PPE you are using, what kind of instruments you're using. So autoclave, all this equipment. There are a lot of active BSL-3s in India, like in AIMS. There is one they're building new in Gujarat. So this cannot happen without funding, but there has to be a mindfulness of the how funding is dependent on the local regulatory environment. But however, in India, the PPEs, it's comparatively priced equitably compared to US. So it is very much possible that high impact research of BSL-3 is possible in the countries like India.
That gives us a strong perspective on infrastructure. So, moving from facilities to experimental models, how do organoid systems compare with traditional in vitro and animal models in terms of reliability, scalability, and translational relevance?
Yeah, that's a good question. Is a very timely, platform because animal work is very expensive. It is now also adding to your first question. The high throughput it reduces because of animal and the expertise required to animal handling and all those is very high. So for organoids, it provides a system in the dish, as I explained in my talk, and you can have high throughput with one organoid, with one small piece of tissue that you get from patients. You can have like 50 organoids, and then that you can use that for research. So each organoid of those almost acts as one animal. So it's a very cutting edge technology given the research environment we are in, and it also reduces the cost of the research.
Okay. Thank you for that clarity. So, building on this comparison, let us now explore how these two powerful approaches can complement each other. So, in your experience, how effectively can BSL-3 lab capabilities be integrated with organoid technology to accelerate the drug discovery and vaccine development?
Yeah, so this is an extension of my answer for the first question. Handling animals in BSL-3 is very complicated. First, it requires a big expertise of animal handling who are able to handle the animals in both the environments. Second is that it's more expensive and it is more time taking. So keeping all these three in mind, having organoid research inside the BSL-3 is the best fit.
Okay, that's very insightful. So, alongside innovation, safety and ethics remain critically important. So, my next question is, what are the key biosafety and ethical challenges researchers must address when working simultaneously with high-risk pathogens and human-derived organoids?
Yeah, so I think there are two separate questions. For BSL-3, the ethical is, and the main hurdle is training. So when somebody is interviewed, whether they are fit for biosafety level 3 labs, there has to be a psychological analysis as well, because most of the biosecurity issues that have been happened in the past is because of one of the lab researchers being involved in that. Right, so somebody's funding got canceled and they got upset, and so they decided to send anthrax letters to the whole world, right? To whole US. You guys know that. So I think the ethical starts with the mindset. So checking the mindset when anybody is hiring a researcher for BSL-3 is very important. And for organoids, I think for organoids and BSL-3 both, it's very important to have hands-on training and to be successful in research.
Okay, thank you for highlighting those considerations. So, looking ahead, it would be interesting to understand where the field is heading. How do you envision organoid technology evolving in the next decade, particularly in the context of emerging and re-emerging infectious diseases?
Yeah, I think this is a very timely platform for emerging diseases. We are seeing H5N1, we are seeing monkeypox, we are seeing so many pathogens just coming out and outbreaks, which are kind of alarming for all of the research community and in general. So having organoids and already developing those models, those disease and dish models to be prepared for these kind of pandemics and future pandemics for this pandemic potential organism is very important. And the organoids offer a platform where you can do this in advance in a minimal cost. So that's a good question. Thank you.
Thank you. So, what skills and competencies should students and early career researchers focus on if they aspire to work in the high containment labs and advanced biological modeling systems?
Yeah, the skills are like at undergrad level or graduate level, read a lot of relevant publications, try to understand, first of all, the basic foundational concepts of all of these. The second is know more about what you want to do. It might be a very important thing in general, but are you really passionate about this research? That is the most important thing to figure out. And then, of course, once you know that you're interested in organoid or BSL-3 or any particular immunological studies, so then, contacting the right professor, then going in their labs and learning all the technologies is very important.
Okay, very valuable guidance indeed. So, finally, considering the global nature of infectious diseases, given the lessons from recent pandemic, how important is international collaboration in maximizing the potential of BSL-3 labs and organoid platforms for global health preparedness?
Yeah, it international is very important because many of the mycobacterial tuberculosis talk that I talked about is all happening. It's impacting countries like India, and also collaborating with international collaborators to know and stay updated with upcoming technologies is very important. So like microarray started in US 10 years before then it started in India. So having that cross collaboration decreases the gap between the technologies that is being transferred from one country to another. So knowledge transfer, training transfer, and technology transfer is very important and that happens only with collaborations.
Okay, thank you. One question is from Ms. Fatima Nakvi. Her question is, what are the biggest challenges in maintaining air filtration, HEPA, systems, particularly when dealing with aerosol generating procedures?
Yeah, so HEPA filters is not a big challenge because you can change it every other year. And then there is a testing done with HEPA filters. There are companies which we hire to do the testing for the integrity of HEPA filters. And they do certain testing for HEPA filter integrity. And if the test comes out as negative, that means the integrity of the HEPA filters are lost, then the filters need to be replaced. And mostly the HEPA filters replaced every 5 years, depending on the environment. If it's a polluted environment, then it has to be filtering a lot of uh, pollutants, so it causes the HEPA filters to damage faster. And however, there are cases like that HEPA filters are fine for 10 years as well. So really depending on the quality of HEPA filters that you purchase to install, and regular testing of HEPA filters are the two main key things for HEPA filtration.
Thank you, ma'am, for that answer. Dear participants, do you have any questions? If yes, so please you can unmute yourself, or you may also type them in the chat box. Yeah, sir, please go ahead.
Yeah. Dr. Jayashree, thank you very much. I enjoyed your talk. I just want to ask, because most of our students are, UG and PG, so those are interested to do their master or PhD at Stanford. Maybe you can elaborate what they need to do if they wish to come to Stanford for PG program, MSc or PhD.
Yeah, I mean, that's a good aspiration to have. Most of the researchers go on to do PhD, right? And so how to go about it is the best thing is to go online. Stanford also has a PhD entrance interview and exam. So once you have the interest, like I said earlier, interest is very important. So whichever program or research you're interested in, because PhD is a long drawn process, right? It's 5 to 6 years. And so you have to be passionate and interested in that topic. So first find the topic of interest on the website. Now, because of the internet, we all are connected. So once you find the topic of your interest and the investigator, the principal faculty, then you contact them. If you contact, honestly, if you contact 20, 30, at least you will get 1 or 2 replies. So it's like the matter of in around US or in Europe, if you find something that you really like to pursue in your PhD, if you find, say, 10 or 20 professors, email all of them and at least you will get 1 or 2 replies. So be constant, don't get disheartened, I would say, because we all want to go somewhere, but many times it doesn't happen. But India in itself is also a very growing platform for research, and don't discount India as a platform for doing research. It has a lot of resources and very good faculty. But do explore Stanford and other universities in US. There are like thousands of universities in US, and all of them are good and great. So I would just suggest to contact cold email. It's called cold email. Email them, and if you don't get reply, follow up. Email more professors. Because of the internet, we can keep emailing without any much charges. So I would suggest to keep going and contacting the professors. Write a good CV. Try to bolden your profile by involving in research. There are summer programs that you can go to AIMS, Bhabha Atomic Research, and many industry-driven research you can do in your undergrad and postgrad, which will enhance your profile. So once you have a profile where you're showing that you're also doing interesting research, the professor would be interested in, replying back to you. So having a good CV with a lot of research experience in the lab or in the industry within India, and then to showcase that you're capable of doing that is one of the ways to proceed in this PhD aspirations.
Yeah. Thank you. The second question is in sense of getting fellowship or scholarship. So maybe you can share your thoughts. So for the benefit of students, getting fellowship or scholarship for doing MSc or PhD.
Yes, there are lots of scholarship because I studied masters in US and I also studied in India. There are a lot of CSI scholarships, the CSIR scholarships. Once you get into the program, so then you can apply for those scholarship. DBT has a lot of scholarship. So look into those scholarship. For US, once you get into a master's program, there is a GRE, you have to write the GRE too. Once you get into master's program, there are no scholarships for international students, but you can get a teaching assistantship or a research assistantship, which will cover the fee of your stay and your lodging and your studies. So there are a lot of scholarships as well and once you're in PhD program, then also most of the labs support the PhD students. Very few have to actually support themselves. So you get a TA or an RA research in the US. In India, there is also a similar kind of thing. When I did graduate from JNU, there are a lot of research assistantship offered by the professor and also by CSIR and DBT. So those are some of those. So tell me, Professor, how do students get into your program of the university? What is the process? Is there an exam or there is a 12th grade that determine it? How do you do it?
I mean, at our Center for Scientific Research and Development, we have 4 programs: BSc Biotechnology, BSc Microbiology, MSc Microbiology, and MSc Biotechnology. So these are not professional courses by the definition of the courses in India. So they don't need to go for any entrance exam. So as long as they have the prerequisite, for example, for BSc biotechnology or microbiology, they just need to have a biology subject in 12th, biology, chemistry, mathematics, I mean these basic sciences, and they can get admission for the UG program. But in case of MSc, they should have a BSc degree in botany or zoology or biotechnology, any biosciences. So there is no any entrance exam at the moment. Yeah.
Okay, this sounds good. Thank
Yeah.
you.
Yeah. Aisha, you can take over.
Okay, sir. Ma'am, we have another question. Uh,
Doctor,
yeah, sir,
I have a
please.
one, Dr. Sushri. Actually, I want to ask you one thing because I am Dr. Hussain, the faculty of CSRD, and I have worked on the Mycobacterium tuberculosis. I just want to know about, please, could you tell our students about the latent TB and the drug which is given for the latent TB tuberculosis, and so that the students can boost their immune system. Could you please tell our students about the latent tuberculosis?
Yeah, so in India and a lot of other countries surrounding India, and latent tuberculosis is quite
Because our
prevalent.
India is a high burden TB.
Yeah, so the latent tuberculosis is quite prevalent, and actually there is no requirement to treat latent TB because latent TB can stay in the body for ages and nothing can happen. I mean, uh, there is a choice whether you can treat latent TB. You can go to the doctor. I'm not a doctor, so you'll have to consult the doctor. But as far as research is concerned, the isoniazid and other drugs are given for a long period of time to get rid of latent TB. But for knowing what to do to cure latent TB is something that you have to reach out to your primary care doctor. Yeah.
Okay.
Thank
Thank
you.
you, ma'am. So, esteemed participants and fellow students, this has indeed been a remarkable session. I extend my heartfelt thanks to Dr. Garyan for her valuable time and inspiring insights. I would also like to thank all participants for their active involvement during this session. Now I hand it back to our MC, Mr. Uttam. Over to you. Thank you.
Thank you, Aisha, for skillfully moderating the session. To move forward, I now invite Siddharth Singh to deliver the vote of thanks on behalf of the entire organizing committee. Mr. Siddharth, over to you.
Thank you, Mr. Uttam. Good evening, everyone, respected dignitaries, esteemed faculty members, and dear fellow students. I am Siddharth Singh, a proud BSc Biotechnology student from the Department of Biotechnology and Microbiology, Center for Scientific Research and Development, People's University, Bhopal, India. It is both an honor and a privilege to propose the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development. As we conclude today's enriching session of the BioConnect webinar series, first and foremost, we extend our heartfelt gratitude to our distinguished guest speaker, Dr. Jayashree Garyan, for graciously taking time out of her busy schedule to be with us today. Her insightful presentation on from containment to cure, the critical role of BSL-3 labs and organized in infectious disease research has been truly enlightening. We sincerely appreciate the valuable knowledge and perspectives she has shared with us. We also express our sincere thanks to the Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, for his gracious presence and encouraging opening remarks. Sir, your continued support and guidance mean a great deal to us. Our heartfelt appreciation goes to all the respected dignitaries who joined us today. Your presence has truly enriched this event. We extend our deepest gratitude to our mentor and guide, Professor Dr. Subhash Bhore sir. Your vision, guidance, and constant support have been the driving force behind this webinar series. Your mentorship continues to inspire and empower us. We would also like to thank our moderator, Ms. Aisha Jamir, for conducting the session so effectively and ensuring its smooth flow. I would like to take a moment to highlight the essence of the BioConnect webinar series for the students by the students initiative. It stands as a testament to how passionate learners can achieve great heights when given the right platform to explore, question, and grow. This series not only connects us with global experts but also fosters a vibrant academic environment driven by curiosity and collaboration. To our enthusiastic audience, thank you for your active participation, engagement, and insightful questions. Your involvement truly added value and energy to today's session. Most importantly, we want to record our sincere appreciation to the management of People's University for their unwavering support and encouragement. Your belief in student-led initiatives like the BioConnect webinar series inspires us to dream bigger and achieve more. With this, I now hand over the proceedings back to our EMC, Mr. Uttam Singh. Over to you. Thank you.
Thank you, Siddharth, for expressing our sincere gratitude to our expected guest, our expert speaker, and esteemed participants. Dear participants, your feedback is extremely important to us. We kindly request everyone to take a moment to fill out the feedback form, the link to which has been shared in the chat box. Your valuable suggestions will help us improve the quality and relevance of our future webinars. Now, we will wait for 2 to 3 minutes to allow participants to complete and submit their feedback. We hope you have had a moment to share your feedback. Please note that your e-certificates will be sent to your registered email addresses shortly. With that, we come to the close of this insightful session of the BioConnect webinar series. We look forward to bringing you many more engaging sessions that connect science, society, and students. I am Uttam, signing off on behalf of the CSRD student team at People's University, Bhopal. Thank you all for joining us today. Wishing you a pleasant and inspiring evening.
Thank you all. Bye bye.